I want you to imagine, just for a second, that you are trying to find a very specific microscopic needle in in an impossibly large continent-sized haystack.
Oh, wow. Yeah, that's a lot of hay.
Right. And normally, to find it, you'd have to sift through the hay by hand, piece by piece. And in the pharmaceutical world, that sifting takes years. I mean, it costs billions of dollars.
It really does. It's an agonizing process.
But what if, instead of searching blindly, you could just, press a button and a machine on your desk prints a custom, perfectly shaped magnet that just reaches directly into the hay and pulls out exactly that one specific needle?
I mean, it sounds completely like science fiction, right? But that analogy, it perfectly captures the bottleneck we've been stuck in with modern drug discovery. We spend decades and literally fortunes just trying to find molecules that will perfectly bind to specific disease targets in the human body.
And, well, changing that reality is the mission of today's deep dive. We are unpacking this really fascinating research paper from ByteDance Seed, and it's titled 865.2026.04.10.7761613v1.full.pdf. It introduces this AI model called Protenix V2.
Yeah, Protenix V2 is quite the leap.
Honestly, looking through these notes, it feels like this model is entirely rewriting the rules of how we predict and design biomolecular structures. It's essentially printing that magnet we just talked about.
It's a profound shift in how we approach biology altogether. We are watching the field move from a world of physical trial and error in wet labs to computationally generating highly specific, viable solutions on the very first try.
Okay, so let's trace exactly how this model pulls this off, because the methodology is just wild. We'll start by looking at how this AI predicts the microscopic shapes of biological components.
Like the prediction phase.
Exactly. Then we'll explore how it uses that site to actually design brand new antibodies from scratch. After that, we have to look at the reality check.
Oh, making sure they actually work outside a computer.
Right. Making sure these digital AI designs can survive as physical drugs in a human vein. And finally, we'll look at the physics check, which ensures the AI isn't just, hallucinating impossible chemistry.
It's an ambitious roadmap, for sure, but you really have to look at all those layers to understand the magnitude of what Protenix V2 is actually doing.
So before you can design a custom key, which in this case is your antibody drug, you have to perfectly understand the microscopic shape of the lock you are trying to open.
Yeah, the lock being the antigen.
Right, the antigen or the disease target. And if your key doesn't fit the lock exactly, the drug just doesn't work.
That lock and key fit is everything. And to measure how well an AI predicts the physical interaction, scientists use a metric called a DOQ score.
DOQ, right. Can you break that down for us?
Sure. This is crucial because it's not just asking if two proteins bump into each other in a digital simulation. DOQ measures the geometric precision of the interface.
It's like how tight the fit is.
Exactly. It scores how perfectly the atomic grooves of the antibody mesh with the ridges of the antigen. It's looking at distance, angles, physical overlap. A score over 0.23 is considered medium or acceptable quality.
Okay, 0.23 is the baseline for acceptable.
Right, and a score over 0.8 is extremely high precision. That's essentially an exact physical match down to the atom.
Which brings us to the actual data for Protenix V2. When I was reading through the benchmarks, the numbers they jumped out immediately.
They really do. I mean, they tested it across 3 major antibody benchmark collections: KX-meter AB, FoldBench AB, and AF3AB.
So they threw a lot of different tests at it.
Oh, absolutely. And at that medium DOQ threshold, Protenix V2 achieved up to 13-point gains in success rates over its predecessor, Protenix V1.
Up to 13 points. That is a massive jump for a single generation update.
And those gains hold up in the strict high-precision regime as well. The researchers compared it against major state-of-the-art baselines like AlphaFold3, Boltz-1, and OpenFold3.
The heavy hitters.
Exactly. But the most important takeaway isn't just that it's slightly more accurate. It demonstrated a massive leap in what the paper calls sampling efficiency.
Okay, here's where the mechanics of this get really interesting. To understand sampling efficiency, we have to quickly explain what an inference seed is.
Right, the seed is key here.
Yeah, because when an AI generates a biological structure, it doesn't just pull it from a database. It starts from a point of mathematical randomness, a seed, and iteratively refines it into a shape.
It sculpts it. basically.
Right. And historically, because this process is so incredibly complex, you have to run the model hundreds or thousands of times using different seeds, just hoping one of them eventually folds into a good result.
In the past, generating like 1,000 seeds to find one highly accurate, usable prediction was standard operating procedure.
But the data in this paper shows that Protenex V2's performance using just 5 seeds actually surpasses the previous model's 1,000 seed results.
Just 5. It's wild.
It reminds me of someone throwing darts blindfolded. If you need 1,000 throws to finally hit the bullseye, you aren't a skilled player. You're just guessing by sheer volume.
Right, it's just probability at that point.
Exactly. But if you hit the bullseye in just 5 throws, you aren't guessing anymore. You have guided, algorithmic precision.
And the real-world impact of that efficiency is immense. I mean, it saves massive amounts of computational time, money, and energy. If you cut down the required attempts from 1,000 to 5, it means researchers can process vastly more disease targets in a fraction of the time.
Which speeds up everything.
Oh, totally. We should also note a quick structural detail regarding the model's validity. The researchers implemented a strict training data cutoff of September 30, 2021.
Okay. That's essentially a firewall, right? To prove the model isn't cheating.
Exactly. It guarantees the AI hasn't secretly studied the answers to the test. It proves the model isn't just, regurgitating memory structures from recent lab discoveries. It is genuinely learning the underlying physics and predicting entirely novel structures.
Okay, let's just pause here and do a quick bullet summary for you, the listener, to lock in what we've covered in this first section.
Good idea. So, bullet 1. Prediction accuracy is massively up. We're seeing up to 13-point gains across major benchmarks.
Bullet 2. Computational waste is massively down. Protenex V2 hits the target using just 5 seeds instead of 1,000. It's precision, not guessing.
And bullet 3. A strict 2021 data cutoff proves it's actually doing the math, not just memorizing the answers.
So just to make sure I'm wrapping my head around this foundation, we aren't just getting a slight bump in predicting how the lock and key fit together. We are cutting out 99% of the computational waste and hitting the precise geometric fit in just a handful of tries.
That's the core of it, yeah.
It really makes you wonder, if you, the listener, had a tool that could predict exactly how any 2 physical puzzle pieces in the universe fit together on the very first try, what kind of impossible problem would you point at first?
Because once you can visualize that lock perfectly, you don't just have to search for a key. You can start forging one from scratch.
Which takes us to the next massive leap in this paper. We know the AI can see the biological lock, but can it build a key that has never existed in nature? The researchers referred to this capability as zero-shot antibody design.
Yeah, the term zero-shot needs a bit of context to appreciate how disruptive it is.
Please, break it down for us.
So, in traditional drug discovery, if you want an antibody for a new disease, you often inject an animal, like a mouse, or even a llama with the antigen.
A llama? Really?
Yeah, llamas and alpacas are huge in this space. Then, you wait for its immune system to react. You extract the antibodies it naturally produces, and you spend years in a lab tweaking and mutating them to make them stronger or safer.
That sounds incredibly tedious.
It is. But zero shot means the AI skips the animal, skips the waiting, and skips the lab evolution entirely. It computationally generates a brand new functioning binder on its very first attempt, with no prior training on that specific target.
It just dreams up a microscopic structure and it works. And they tested this generative ability on different types of antibodies. There are 2 acronyms we should define here, VHH and MAB.
Right. Let's cover VHH first.
So VHH refers to single-domain antibodies, which are actually derived from the immune systems of camelids, like those alpacas we mentioned. They are tiny, nimble, and great at squeezing into hidden crevices of a disease target.
Very useful for hard-to-reach places.
And then you have MABs, or monoclonal antibodies. These are your traditional, heavy, full-length, Y-shaped antibodies that our own bodies produce. Protenex V2 was asked to design both formats.
And the results on standard soluble disease targets were exceptional. Across the board, they achieved a 100% target level success rate.
100%?
Yeah, meaning for every single soluble disease target they tested, the AI successfully generated at least one working, physically viable drug candidate.
But the nuance of how the AI does this really comes into focus with the AMBP case study. AMBP is a specific protein target. When the scientists asked the AI to design an antibody for one specific surface patch of AMBP, the model struggled a bit. It had like a 4% hit rate.
Which is pretty low.
Right. But when they pointed the exact same AI at a different patch on the exact same protein, the hit rate skyrocketed to 48%.
This highlights a crucial mechanism in biomolecular design. Epitope choice, meaning exactly where on the target's topography you choose to attach your drug, dictates the computational difficulty.
Like picking the right landing zone.
Exactly. Proteins are not smooth spheres. They are jagged landscapes with deep pockets, flat planes, and slippery curves. The AI's success is highly sensitive to the physical shape of that landing zone. And while soluble targets are a great proof of concept, the true boss level of drug discovery is the GPCR.
GPCRs, cheap protein-coupled receptors. Okay, for anyone not deeply entrenched in biology, imagine a door on the surface of a human cell. The GPCR is the door handle.
I like that analogy.
If a drug molecule can grab that handle, it sends a chemical signal inside the cell to change its behavior. But trying to grab a GPCR is like trying to grab a tiny, slippery, wobbly handle on a door that keeps violently opening and closing. They're notoriously difficult to target.
And the reason they are so difficult is because they are membrane proteins. They're embedded within the fatty exterior of the cell.
So they're mostly hidden.
Yes. The exposed surface area sticking out of the cell is tiny. And because it's constantly transmitting signals, the protein is highly flexible and constantly changing shape. Conventional antibody discovery methods fail here constantly because the target literally won't hold still.
Yet, Protenex V2 took on multiple GPCR targets. And it did so on an incredibly tiny experimental budget. In the lab, they only tested between 16 and 30 AI-generated designs per target.
Which is almost absurd.
Right. In the pharmaceutical world, testing 30 designs is nothing. Usually, you are screening tens of thousands of molecules just hoping one sticks.
Yes, the testing budget is minuscule. And despite only testing 30 options, the nimble VHH formats achieved hit rates ranging from 16% all the way up to 88% on these wobbly door handles.
That's incredible efficiency.
And the heavier MAB formats hit up to 50%. But there's one specific measurement we really have to highlight here. For a GPCR target called GPRC5D, the AI designed a binder with a KD of 112 picomolar.
Okay, let's translate that because that number is staggering. KD is the dissociation constant. It measures binding affinity, basically how tightly the drug holds onto the target.
The grip strength.
Right. The lower the KD number, the tighter the grip. Picomolar is insanely tight. It means the AI designed a molecular claw that grabs onto this slippery, shifting door handle and absolutely refuses to let go, even under microscopic physical stress.
And it achieved that binding strength computationally in zero shot. And researchers only had to test a few dozen tubes to find it.
Okay, time for our next 5-minute recap for you. Bullet summary time.
Let's do it. Bullet 1. Zero-shot design works. The AI is dreaming up functional antibodies on the first try without relying on animal immune systems.
Bullet 2. Epitope choice matters. Where you am on the protein landscape completely changes your success rate, like we saw with AMBP jumping from 4% to 48%.
And bullet 3. Even the hardest targets, those wobbly GPCR door handles, are yielding to AI, and they're doing it with tiny testing budgets of around 30 tubes, hitting picomolar affinities.
So think about the efficiency here. I'm asking you, the listener. If we can generate valid drug candidates by testing 30 things instead of 30,000, how does that change the timeline of human medicine?
It accelerates the timeline by years, potentially decades, over a broad spectrum of diseases. But we have to temper that excitement with biological reality. Binding tightly to a target inside a digital simulation is really only half the battle.
Which brings us to the reality check, because it is fantastic if a molecule binds tightly in a computer model. But if you manufacture that exact drug, put it in a glass vial, and it instantly falls apart at room temperature, it's completely useless.
Right, it's literally garbage.
It can never be shipped to a hospital, let alone survive in a human body. This survival concept is called developability.
Developability is essentially a series of harsh physical stress tests. You can think of proteins like incredibly complex origami. If the origami unfolds or if the paper sticks to itself, the function is lost. The researchers focused on 3 main pillars to ensure these AI-generated proteins behave like stable, real-world drugs.
I love how they laid out these stress tests. Let's look at them conceptually as the melt test, the clump test, and the friendly fire test.
That's a great mnemonic to remember them by.
First up, the melt test. The scientific term is thermostability, or DSF. Proteins are held together by delicate atomic bonds. If the environment gets too warm, do those bonds break? Does the drug literally melt and unfold?
Right. And to pass this, they want the apparent melting temperature to be above 60 degrees Celsius. The Protenex V2 designs had a 100% pass rate. Every single design they tested maintained its structural integrity. They are rock solid.
100%. That's crazy. Then you have the clump test, or self-interaction. They call it ACS-INS. Antibodies sometimes have sticky patches on their exterior. The risk here is, do these molecules accidentally stick to each other instead of the disease?
Which is a huge problem.
Yeah, if they do, they clump up into a useless, gooey gel, which is the last thing you want injecting into your bloodstream.
Absolutely. But the AI design scored a 98.5% pass rate on the self-interaction test. They remain dispersed and active.
And finally, you have what you call the friendly fire test. The official term is polyreactivity, checked via BVP ELISA. This asks a critical safety question. Does the antibody specifically and uniquely attack the disease, or does it accidentally stick to random, healthy human cells?
You definitely don't want your immune system getting confused and attacking healthy tissue. And the pass rate here was a stellar 93.3%.
So the AI isn't just making pretty digital art. It is engineering physically robust, highly specific medicine that survives the real world.
It's passing the physical gauntlet.
But here is where the research takes an amazing, almost philosophical turn regarding the role of humans in this process. Let's look at the VEGF-A case study.
Ah, yes. VEGF-A is a complex disease target.
Yeah.
The researchers took a pool of about 300 AI-designed drug candidates for it. They had 2 different AI algorithms evaluate that pool and pick their top 30 favorites.
Okay.
Then they brought in a human biological expert, gave them the exact same pool of 300, and asked the human to pick their top 30 favorites.
The ultimate human versus AI showdown. And the mechanics of how they chose are so revealing. The AI algorithms methodically found the molecules with the absolute strongest binding response. They found the deepest pocket on the target and just maxed out the strength stat.
Poor optimization.
Right. But the human expert, the human, found 7 completely unique binders spanning a much wider variety of structural clusters. And those 7 human-picked binders, the AI had completely ignored them.
This perfectly illustrates the difference in how we evaluate good design. The AI models are relentless optimizers. They concentrate heavily on a small number of highly productive, very strong binding modes. If one pocket gives the best grip, the AI puts all its chips on that pocket.
All its eggs in one basket.
Exactly. But the human expert looks at the entire chemical landscape differently. The human brings strategic intuition. They select candidates that might have a slightly weaker initial grip, but they offer entirely different angles of attack.
Because what if the virus mutates and that deep pocket disappears? The AI's perfect drug is suddenly useless. The human is looking at the board and thinking, we need backup options. We need to attack from the side, from the top.
It's about resilience.
Yeah. The AI is the pure strength optimizer, but the human is the creative explorer looking for weird, unique solutions to ensure resilience.
It shows that in drug discovery, human intuition and AI optimization are fundamentally complementary forces, not competing ones.
Okay, let's lock in section 3 for you with another quick recap.
Sure. Bullet 1. The designs survive the real world. They pass the melt, clump, and friendly fire tests with over 90% success rates across the board.
Bullet 2. AI and humans look at problems differently. The AI optimizes for brute strength, but humans optimize for diversity and strategy.
Which means, bullet 3, we still need human experts. The AI didn't replace them, it just gave them a better pool of candidates to strategize with.
So we know these designs can survive the real-world stress tests. And we know human experts still provide immense value. I have to ask you, listening to this, does it comfort you to know that human intuition can still spot valuable solutions that a super advanced AI overlooks?
I know it comforts me. The machine is a tool to augment our expertise. But to truly trust that tool, we have to look at its fundamental grasp of reality, which leads us to the final hurdle: chemical plausibility.
Right. Up until now, we've talked about massive protein structures like antibodies, but modern medicine also relies heavily on small molecules, or ligands. Think of your standard pill form medication. And when designing these tiny molecules, the AI sometimes acts less like a structural engineer and more like a science fiction artist.
That's a perfect analogy. We have to remember that AI is fundamentally a statistical model. It guesses where atoms should go based on vast patterns in its training data. It is not inherently a physics engine.
It doesn't actually know physics.
Right. So sometimes it will predict a small molecule structure that looks incredibly effective from a distance, but when you zoom in to the atomic level, it completely violates the laws of physics.
So without strict rules, the AI was basically drawing M.C. Escher's staircases. You know those optical illusions where the stairs loop infinitely?
Oh yeah, the impossible stairs.
Right. It looks cool on paper, but you could never actually build it with wooden nails because gravity and geometry won't allow it. The AI was trying to build carbon structures that simply cannot exist in physical reality. It might overlap two carbon atoms or twist a bond in a way that would instantly snap in the real world.
Specifically, it was failing on geometric constraints, like planarity, meaning atomic centers that physically must be flat, were being drawn bent.
Like bending a flat board.
Exactly. To fix this hallucination problem, the ByteDance team implemented something called TFG, or Training-Free Guidance.
How exactly does TFG stop the hallucinations?
You can think of TFG as a software patch that actively installs the fundamental laws of physics into the AI's generation process. During the moments when the AI is trying to position an atom, TFG acts as a supervisor.
Watching over its shoulder.
Right. It enforces strict geometric rules regarding torsional angles, atomic distances, and bond lengths. If the AI tries to twist a molecule in an impossible way, TFG mathematically blocks that move and forces the design back into the realm of physical reality.
It's giving the AI an architectural building code it is mathematically forbidden from violating. And the data shows this strict supervision works. They ran the designs through incredibly harsh validity criteria. They used this thing called pose busters. And with TFG installed, our 10x V2's success rate in generating chemically realistic, highly accurate ligand poses jumped to over 60%.
60.46% to be exact. It grounds the AI's imagination.
But the true culmination of this entire paper, the ultimate test of everything Proteinix V2 can do, is the SARS-CoV-2 case study. They didn't just ask the AI to design a binder for one specific version of the COVID-19 virus. They set an incredibly high bar, a multivariant design.
This part of the paper is wild.
Right.
They gave the AI the microscopic target structure of the original COVID-19 prototype strain, but then they also gave it the structure of the highly mutated Omicron strain.
The B.1.1.529 strain.
Yes.
Yeah.
They essentially told model, design one single drug that effectively attacks both of these distinct viral structures.
Which is an incredibly difficult engineering problem because viruses mutate specifically to escape these kinds of drugs. For instance, the Omicron strain has a very specific mutation called Q493R.
Ah, the Q493R mutation.
Yes. What that means is the virus replaced a small amino acid with a much bulkier one, introducing what we call steric hindrance. It's basically physical clutter that ruins the binding ability of older drugs.
It's like the virus put a padlock over the door handle.
Exactly, but the AI somehow solved it. Out of just 4 designs they tested in the lab, 2 of them successfully bound to both the original strain and the mutated Omicron variant at an anomalar affinity.
What's truly fascinating is how the AI solved that padlock problem. It built compensatory mechanisms into its drug design.
Oh, so?
When the drug encounters that bulky Q493R mutation on the Omicron virus, the AI's design molecule actually physically reorients one of its own amino acids, moving it out of the way of the clutter to establish a brand new interaction with a completely different part of the virus.
That is incredible. It literally adapts its molecular grip based on the specific mutation it encounters. It's an intelligent, flexible claw.
It is.
Which really shifts how we think about fighting disease. If we can engineer physics-bound molecules that dynamically attack multiple viral mutations at once, we are finally moving away from just playing defense and starting to play offense against viral evolution.
For decades, pharmacology has been entirely reactive. A virus emerges, it mutates, we study the mutation, and we try to build a new defense. This technology points toward a proactive future. We can use models like Proteinix V2 to anticipate evolutionary escape routes and computationally block them before they even emerge in the population.
Okay, time for our final quick recap for you before we wrap up.
Here we go. Bullet 1. TFG is the physics police. It stops the AI from drawing M.C. Escher staircases and ensures the molecules can actually exist.
Bullet 2. It passed the cross-variant test. The AI successfully designed one single drug that attacks both the original COVID strain and the highly mutated Omicron strain.
And bullet 3. It defeated steric hindrance, that molecular clutter, by designing a flexible claw that adapts its grip.
So my question for you here, if we can engineer physics-bound molecules that attack multiple viral mutations at once, are we finally moving from playing defense against viruses to playing offense?
It certainly feels like the tides are turning.
So let's bring this entire journey full circle to understand the weight of this research. Proteinix V2 isn't just a software update that makes things run a little faster. It is a fundamental bridge between digital blueprints and physical viable cures.
Absolutely.
We started with predicting shapes. seeing how the model cuts through computational waste to find the perfect lock-and-key fit with incredible efficiency. Then we saw it act as an architect, zero-shot generating brand-new antibodies that conquer the hardest, flipperiest GPCR targets in biology using minuscule testing budgets.
And it did it so well.
We verified that these AI dreams can survive the harsh physical realities of melting and clumping, while proving that human creativity is still vital for finding diverse strategies. And finally, we saw it use strict physics guidelines to design an adaptable COVID-19 drug that outsmarts viral mutation.
It really represents a maturation of the entire field of computational biology. We are moving past the theoretical phase of asking, can an AI draw a pretty protein? And we were fully entering the practical phase of asking, can an AI reliably design a safe, manufacturable, high-affinity drug? And the data in this paper strongly suggests the answer is yes.
Which leaves me with one final provocative thought for you to mull over. We started this deep dive by talking about the agonizing, years-long process of finding a needle in a haystack. Today, when a new virus emerges, it takes years to design, test, and validate a drug to fight it.
Years of lost time.
But if an AI, like Proteinics V2, can design a cross-variant, adaptable binder on the very first try, if it can verify the physical fit using only 5 computational seeds, and if we only need a tiny batch of 30 test tubes to validate it in a lab, are we approaching a very real future where a new unknown pathogen is sequenced in a lab on a Monday, and its precise, physical, AI-generated cure is computationally solved, verified, and sent to manufacturing by Tuesday?
Given the trajectory of this data, I'd say that future seems far closer today than it did yesterday.
Thank you for joining us on this deep dive. Keep questioning the incredible science shaping our future, and we'll see you next time.